Alright students, let me make this very clear that I have turned the page now and this is a fresh page, right? It's not the old one, this is a fresh page. If you can see, the heading is changed here. It is Anatropous ovule. I told you that we will be getting into detail about what is an Anatropous ovule. I expect that by this time you understand that ovule is a megasporangium. Megasporangium. Right? An ovule is a megasporangium, and when it is immature, when it is mature, this I'll be telling you in this session. Right? As of now, the diagram that you are watching, this diagram is a matured Anatropous ovule since it contains this central body. If you can see that I have labeled this central body as an embryo sac. That means whenever this embryo sac is present inside the ovule, it is matured enough to undergo fertilization. There is a reason behind that, because embryo sac contain one special cell which is known as egg cell, and I told you that this egg cell is known as megaspore. So the moment you have a megaspore within the ovule, that means the ovule is matured enough and it is ready for fertilization. Alright, what are the key features for Anatropous ovule? First of all, you see the labelings. This is one ovule and an ovary may contain hundreds of such ovules within it. Right? We are magnifying only one ovule so that you can see the internal details. If you see that an ovule is connected, suppose this is the ovary wall, right? And this ovule is somewhere connected to the ovary wall. So in that case, this connection is known as funicle. This connection is known as funicle. Then you see that there is a notch where this connection is swollen into a whole oval structure. So the point where this connection joins this oval structure, it is known as hilum, right? Whenever you draw this ovule, diagrammatically it is very, very important. Keep it on top priority, right? Because this comes straight away for 3 to 5 marks, a well-labeled diagram of Anatropous ovule. Or they may ask you to draw a megaspore. When you are asked to draw a megaspore, in that case you know that megaspore is present only within this central cell. If you draw the same diagram for that, you will be getting full marks there. So no need to bother about. Whenever you are asked to write, to draw an Anatropous ovule or a megaspore or a megasporangium, doesn't matter, you have to draw one and the same thing. Alright. This is one ovule, and if you can see, I have made 2-3 layers towards the outer side. These layers are known as integuments. We know that when we talk about our body skin and the skin system, we call it integumentary system. That means integument is something which is the outer covering. Right? The outer covering of the ovule is integument. In future, this covering is seed coat. If ever you have seen that chickpea or black gram, the brown coat is the seed coat, which initially was an integument, and after maturity, after fusion, it became the seed coat. Whole oval structure, you divide it into two ends. One is the blunt end, right? This is chelicil end. And the other one is the end with a pore. The opening is there. If you can see, there is an opening here, right? You call this pore as micropyle. So one end is chelicil end, and the opposite 180 degree to chelicil is micropyle, right? Then I have drawn few stuffing cells here. These stuffing cells are nucellar cells. These all cells are parenchymatous in nature and they are responsible for food storage and nutrition, right? In due course of time, in the center is developed this embryo sac. What is the speciality of the embryo sac? It contains 7 cells but 8 nuclei. 1, 2, 3, 4, 5, 6, 7. So 7-celled stage is there. But why 8 nuclei? Because rest of the cells are having one nuclei per cell, one nucleus per cell, but the central one contains 2 nuclei within the common central cell. So in totality, the number of nuclei are 8, but the number of cells are 7. Alright. And then you have to see that what is the arrangement. These 8 cells within this embryo sac are arranged in a particular fashion. You see that on one end, towards the chelical end, there are antipodal cells, 3 antipodal cells. In due course of time, they degenerate. In the center is, 2 nuclei are enclosed within the common cell. You call them polar nuclei. In future, it forms the primary endosperm nucleus, which is a nutritive body. And on the other side, towards the micropylar end, are the 3 cells, out of which the central one, I have shaded it separately. This is your egg cell, the heroine of the story rather. This is a megaspore which will be participating in fusion. This is the female gamete which will be participating in fusion. And rest of the 2 cells are the supporters. They provide nutrition, support, and protection to the egg cell, right? And this is the whole structure of your Anatropous ovule. It seems somewhat like this. Within the ovary, I have drawn one ovule. I have told you that an ovary might contain hundreds of such ovule within it, depending upon the size. One more thing that I would like to clear here. I told you that initially when you draw an ovule, this body is not there. The whole stuff is filled with these nucellar cells all over. The central structure is not there. At that time, the ovule is immature because there is no megaspore within. So no fusion will take place. But in due course of time, this central structure would arise. That means now the egg cell is there and now it is ready to fuse. In that case, you will be calling this ovule as a female gametophyte which contains a megaspore in the center, right? One thing more I would like to make clear. If you see this one was an Anatropous ovule, what will you call this one? I have inverted the ovule, but the content, the logic behind calling this one also an Anatropous. This is also Anatropous. Why? Because I have just inverted, but the logic is that the funicle and the micropyle should lie on the same plane. If both of these structures lie on the same plane, it is an Anatropous ovule. On the contrary, if you see this opposite image, here funicle is at one end and the micropylar end is just opposite to it at 180 degree. Right? So this will become orthotropous here. This will become orthotropous. So you don't have to talk about this as of now. You will be talking about an Anatropous ovule, no matter you draw it inverted or you draw it straight, but the funicle and the micropyle would lie on the same plane. Then it will become an Anatropous ovule. I hope I have made this clear.